get you.
Genetics, just exactly now. Yeah, the film about genetics is now.
Oh, where? Okay. I'm unsure if I have to wait for the organizer to start, but my gut feeling is that I should start because it's 3 o'clock. So yesterday we tried to convince you that genetic variations in everything, and specifically that changes have shaped our genome over the past many thousand years to make of what we are based on functions of a specific library made of 4 magic letters, A, T, C, and G, that combine in a specific way with 20,000 books or genes to make humans what they are. Today I will convince you on the reverse. Genetics is in everything, but everything is not genetics. So I will remind a little bit with a few slides what we discussed yesterday about the major step that shape what we know today as genetics, as a discipline, as a biological concept, and specifically we discussed how this specific project is really the basis that have completely revolutionized our knowledge in genetic and disease over the past 20 years. Once again, as a reminder, we spoke about our genome library that is here, with only about 1.5 to 2% that code for genes, about the same proportion that are extremely conserved, but that may be important in evolution. We spoke about the fact that since we know how to read the genome, the number of disease genes have increased exponentially. So every single day, we know of a new disease genes associated to human disease. You remember this picture? The picture from my 10-year-old daughter. So how geneticists works. They spend time to associate variation in the human genome to a specific picture. Here, this picture could be a disease. This picture could be a trait, being taller, being shorter. This picture could be a shape. What I didn't tell you yesterday is that my daughter told me I know how to do these things grade 1. Why do we need to study that much to combine, to match make? The reality is that it's more complex than that. For example, from time to time, you may have 2 genes, mutation in 2 different genes. In other words, mistakes in 2 different books that would lead to the very same condition. And you may have in the same genes different mutations that lead to 2 different conditions. Or in other words, mistakes in 2 different chapters of the same book leading to 2 completely different diseases. And this makes our work a little bit more complex. It means that one gene, one disease doesn't apply. What are the type of changes that we have that we call mutation or polymorphism? Changes may be silent. So if you may have changes in your DNA, in other words, a letter change in one of the book chapter or in one sentence, but that doesn't lead to any effect. And these ones are called silent mutation. Sometimes you have a change that completely changes the meaning of a sentence. I like banana. If you remove nana, it means you have I like ban. It doesn't mean the same way, right? That's a mischange, missense mutation. So that means a single change that change amino acid. As you remember, the vehicle of messages in the body are proteins. And the proteins are composed of different amino acids. And different amino acids are coded by genes. And this code is made of 3 letters. If you combine ATCG, ATC in a different way, it will lead to one specific amino acid. If you remove the C and replace by A, that's a change, that's a mutation. Sometimes that mutation will change the amino acid. Yesterday we spoke about sickle cell disease. A sickle cell disease is a single change. For example, you have one person that have an A at this DNA sequence. You replace that A by the T, just A and T at this specific loci, and the T by a C at that specific loci. This one doesn't mean anything, neutral, but if you change by a C, a very severe disease called sickle cell disease. This was shown by a very clever scientist called Linus Pauling about 50 years ago. And this was the very first time someone showed that the change of one single letter, remember we have 3 billion of those letters, 3 billion nucleotides. You change only one, it led to a very severe and potentially lethal condition. The reason why this condition is being called the very first molecular disease. And Linus Pauling won 2 Nobel Prizes for that discovery. It was a very, very important discovery. Another type of change is a nonsense mutation that converts an amino acid to a stop codon. Stop codon means the protein stopped to be synthesized, the protein stopped to be produced. Instead of say, I like banana, you say, I like. You like what? So you have a protein that's shorter. That doesn't mean what it was supposed to mean, biologically. Sense mutation is the reverse of nonsense. That means you change a single code, but you have a longer sentence. So you change one specific letter. Instead of I like banana, the sentence is I like banana and apple. It's not what the meaning of the biology was I like banana. You change one letter, is add something else. That's a sense mutation. There is another type of changes that we call French shift mutation. Imagine that for a specific amino acid, let's say for example glutamine, you need A, C, and T, just an example. If you move the A, C, and T and introduce another A, it will be A, C, and T. That means that you have shifted the frame. You will have a protein that will be A, and C instead of A, C, T. So that means the amino acid that will be produced, it will be amino acid that is linked to A and C. Completely different protein. All these changes are changes that happen within the DNA, within the magical letters, the 4 magical letters on the library of life that we call our genome, that we know of. There are some other mutations that we call messenger RNA mutation. It doesn't happen in the DNA itself, but it happens in the next stage of the protein production. You have the DNA, you have one intermediate state that is called RNA, and then you have the proteins. And that change is happening in the second stage. And another changes that is called trinucleotide repeat. For now, all the changes that I have illustrated in the 2 slides before are what we call stable mutation. Stable mutation means that from generation to generation, it will not change. The first time, your child may have the very same, the grandchild will have the very same for the next many generations. This one called trinucleotide repeat is not exactly the same changes per generation. Once again, the biological meaning, I like banana. In this type of mutation, a normal person will say, I like banana. Some people will say, I like banana. You still understand the meaning. Some other will say, I like banana. It's still okay. The only thing that you're going to tell the person to do, see a speech therapy, because you're stuttering. Is it stuttering, right? Okay. Bégayer in French. But let's imagine that you have at the next generation someone where the biology say, I like banana, go outside and come back in the room, Everybody say that guy is crazy. And that's clearly a mutation because you don't know the meaning of the sentence anymore. So from generation to generation, the stuttering become longer and longer and lead to a specific condition. One of those conditions is one of the most ugly genetic condition that we know of called Huntington's disease. Another condition that due to this type of mutation is Fragile X, that is linked to mental retardation, mostly in male. Fragile X syndrome. But having a single changes that lead to a specific disease doesn't mean that that changes will make it all for your disease. This condition, we know it very well in South Africa and specifically in the Western Cape, called familial hypercholesterolemia. This is due to a specific change that disturbs your capacity to process cholesterol. But even within the same family, people will not present the disease the same way because it will now depend a little bit on your diet and your lifestyle. So one of the things to modify your gene expression is environment. You probably all know Michael Jordan, some of you. A very tall American, very famous, probably one of the best basketball players that we have known of. Imagine that Michael Jordan was born in Ethiopia at the time of starvation 30 years ago. Would he have been Michael Jordan? Probably not. He would have been shorter because he would not have enough food to eat. Even though he had predisposition to be very, very tall, he wouldn't have it because there is no environmental factor to allow him to be tall. You may have a change, gene mutation that is there to allow a specific condition, but this can be modified by environment. So even for monogenic disease, this is due to a very single gene. According to how your lifestyle is, you may not present the disease the same way. The same apply for diabetes mellitus. The same could apply for morbid obesity. Because as you know, South Africa is now number 3rd in the world for obesity epidemic. Number 1 in the US, number 2 is Mexico, number 3 is us. So we need now to put it as a priority. Otherwise, we're going to have more and more type 2 diabetes in 20 years. Not even 20 years now already. But in 10 years and 20 years will be a little bit more than that. So environment can be modified. Alzheimer also is another condition where we think probably environment can change the evolution. So in terms of disease, human disease in general, You have those that are called Mendelian, those that are depending on a single mutation. They are very relatively rare. It's about 4 in 1,000 or 14 in 1,000. Those that we call chromosomal, like Down syndrome, 7 in 1,000. Those that we call congenital malformation, like congenital heart defects, about 22 in 1,000. But most of the disease conditions that we all have, all will have, are complex. They are a combination between our genes and our environment. This is high blood pressure. This is diabetes mellitus. It's been shown, for example, that in African city now, 4 an adult out of 10 have high blood pressure. The things that most of them don't know. For those that are measured, they will have high blood pressure. If you move in a room, still in Africa, it's between 1 in 10 or 2 in 10. So clearly our lifestyle in town doesn't allow us to manage our vessels very well. What people call stress, right? Stress and the way we eat. We eat fast, we drink coffee, we smoke, we eat fast again, we drink coffee, we smoke, and we don't sleep. One condition that is mostly cancer, mostly genetic is cancer. Generally cancer is always due to a specific mutation. Now I will introduce a very complex concept called epigenetics. Epigenetics is the changes of your gene expression without changes of the genetic code. I like banana. You have a mutation, I like banana, or I like. You have a mutation, I like banana. Sometime you may have I like banana without a mutation. Strictly no change in your DNA, no change in the letter, no mistakes in the books, but the books cannot read well. So the simple definition of epigenetic is inheritable changes in a gene expression that is not due to the alteration of the DNA sequence. The most obvious case, I don't know, is there a twin in the room? Someone is twin? Someone is twin? No. Monogenic twin, for example, the twin that have the very exact same genetic composition, they don't respond to disease the same way. That's one example of why, because they have the same genetic composition, but their gene expression is very different. This is one typical example. These are actually 2 babies that we saw at birth many years ago. And they are monogenic twins. We tested them genetically. We know they are real twins. They have the exact same genetic composition. But one of them have a very severe malformation at birth. The other one didn't. So clearly that very severe case of malformation is not the result of a genetic changes that is due to a change of the initial DNA. But to stop some of the genes not to express themselves is important for our body. It's important for human development. One example of such is, maybe I'll start from the bottom, is germline tissue-specific genes. For example, we have 20,000 genes. If I want to do a DNA testing in any of us, I may take a blood, that's the way we do it often. I may take saliva, that's the second way. I may take a piece of your hair, third way. I may take a piece of your skin. I will have the very DNA content. But why is that the liver is the liver, the pancreas is a pancreas, a skin is a skin? They have the very same genetic composition, but they don't do the same thing. The pancreas produces insulin. the liver will produce a completely different type of protein. But they have the same genes. Why is that the genes are not doing the same work? The only reason this happens is that this conceptor of this system, God for some, have say in the liver, all these genes that are not there to produce albumin, shut up. Without mutation. It's an epigenetic phenomenon. So it's covered up with something called methyl element, methylation. So they don't speak. Only those that are important for liver function produce something. That will be the same. So it's cell specialization. So without this epigenetic phenomenon, we could not be the beings that we are, because the cell will be doing the very same thing. The second in the middle is happen only in women. Remember yesterday we say 23 pairs of chromosomes. The last pairs of chromosomes is what we call sex chromosomes. The woman will have 2 X's, the male will have 1 X. Is it necessary to have 2 X's to be a normal being? No, otherwise male would be completely abnormal. But they are not. We are not. It means that it is necessary to have 2 X's then in a cell. The answer is no. That means that in all women, there are 2 X's, but 1 is shut down. 1 is not speaking. 1 doesn't express any genes. Otherwise, you're going to have too much production of the same genes if the 2 was expressed. So this silencing of chromosome X in female is absolutely physiological. You need to have it. If something goes wrong, it will lead to a disease. And this concept was described by Mary Lyon. In fact, it's a geneticist, a very famous geneticist, and that concept is called Lyonization, to give credit to her. Female need to stop one of the X to be expressed. Absolutely. And this is a very important mechanism. It's important for gene compensation. The third mechanism where silencing is physiological is a fascinating one. It's called genetic imprinting. To have a baby, you need 2 eggs, 2 germline. One is a sperm, the other one is a ovule, egg. Fecundation, you have again 46 chromosomes, and then you have a normal being. Someone may say, if you put 2 sperms, or if you have 2 eggs in the very same cell, you still have the 46 chromosome, right? A complement of 20,000 pairs of genes. Will you have a normal being? The answer is no. You absolutely need 23 chromosomes from a male and 23 chromosomes from a female to have a normal being. Because there are some genes that will be expressed just because they are from the father. And some other genes will be expressed just because they are from the mother. And the reverse is true. This concept was first described by one of the mentors that I have the privilege to have, Professor Angel, that was the former head of genetic medicine in Geneva. And at the time he described the concept, that was very recent, that was in '83. He described it only in mice, but we couldn't know that this concept will also apply in humans. And a couple of years later, exactly in '91, we have a typical example how this concept may even lead to diseases. I'll come to that a little bit later. So, paternal or maternal differential expression of certain genes is called paternal or maternal imprinting. Because this gene comes from the mom, it will not show up, or it will show up. Because this gene comes from the dad, it will not show up, or it will show up. Very, very fascinating concept. Socially, we need our dad and our mom to grow properly, genetically too. Can changes in this concept is epigenetic through methylation lead to cancer? The answer is yes. And we know now that many cancers, specifically brain cancers, happen because some of the genes are methylated, because they have been shot for many reasons, not because of a specific mutation. And this is many lists. You don't need to know that, but it's just the list of many cancers where we know is not because of a mutation in these genes, but rather to a mutilation in those genes. Now, we will try to illustrate the concept of methylation, which means shortening up some of the gene without any mutation in human disease. In fact, this is the book written by Professor Angel and Antonorakis on that concept. The first one is called Prader-Willi syndrome. Prader-Willi syndrome is a condition that is associated with big size, obesity, mental retardation, and some behavioral changes. These kids will do everything for food. They are obese because they like food, and their behavior all their life revolves around food. So they'll do everything for food, believe me. They will have a little bit of mental retardation, but they will be very clever for food. They will steal food in supermarket. They will put food under their bed. They would like to have a second key of the fridge, because one of the things that we do is that you should have a fridge with a key at home. But they will find a way to have a second key for the food. Everything revolves around food, and they end up extremely big. And this condition is due to a specific deletion, either so one specific missing on shelf number 15 on this row is not there. But you need that this missing happen on chromosome 15 that the child received from his father. Because if that missing happened on chromosome 15 that the child received from the mother, is a completely different condition. I'll talk to the same condition is Angelman Syndrome. The other way is to have in the very same child 2 chromosome 15 from the mom, which is called uniparental disomy. Disomy means 2 chromosomes from the same parent, and in this case is the mom. And the third way is that you may have this methylation on one of the chromosome 15, specifically the one from the dad. But remember this shelf, remember this address, 15Q, like the level of one shelves, chromosome 15. You have the very same changes on the very same 50Q, but this time from the mom, you have a condition that is completely different, called Angelman Syndrome. No obesity, much more severe mental retardation, epilepsy, and again, a very specific behavior. These are very happy kids. They laugh all the time. But the ha is inappropriate. It's not linked to a specific circumstances. So they will arrive to your office, start laughing, go out of the office still laughing. Why? For no reason. In fact, maybe the happiness gene is there. So this was the very first time it was shown that some of these genes that are differentially expressed according to the parents of origin that are located in this specific place on chromosome 15 can lead to specific human disease. Another area where imprinting defects have happened was this in vitro fertilization. As you all know, for a couple that struggle to have baby, from time to time, the baby is conceived out of the uterus and put in the uterus by in vitro fertilization. But it's been shown that some of those eggs that stay in the dish for a few hours, sometimes a few days, before being put back into the womb, the time they spend in the dish changes the methylation pattern of certain genes. So the risk for malformation increase in those kids is 2 times higher. So in fact, in our clinic, fertility clinic, we have to counsel couple that want to go through that process, that the risk of specific some malformation will be higher. But of course, higher doesn't mean 10 times or 20 times. The level of malformation in the general population is about 5%. So if you take a random population, at birth, in 100 kids, 2.5 will have a malformation. But the only study that was perfectly done was done in Australia, and they showed that in kids that was born after in vitro fertilization is not 2.5, is 5%. So it increases the level of malformation. And this malformation specifically, this condition called Beckwith-Weider syndrome, that is associated with very big tongue. One change is that we call omphalocele, and also incapacity to metabolize sugar. These kids are very big at birth, and is due to a methylation of one specific address here. In the general population, this is the proportion, but in kids that are born after in vitro fertilization, this number is much more higher. So I got this like last week. This, my friend, Neil Anshart, is a pediatrician from Baylor College in Houston. And he showed last week that even severe malformation alter the methylation of genes. So that means that some of these kids will not express the gene the same way if they were not malnourished. My other colleague from Rwanda, his name is Mutesa, Dr. Mutesa. He showed that there is a transgenerational effect of trauma on gene expression by studying behavioral abnormality and changes that happen after the drama of genocide in Rwanda. So there is a memory of trauma that can be transmitted from generation to generation. This cannot happen through mutation. It happens only through epigenetics. It happens only through the concept of methylation. There are some other studies that shows that there is also a transgenerational effect of methylation due to alcohol. So if someone has been drinking a lot during this time now, sometime the grandkids may have the effect of that drinking because some of the genes will be expressed differently. This is not due to a changes in the letter itself. It's just due to a change of the gene expression without a changes in the 4 magic letter of our DNA sequence. So to summarize a little bit, 4 magic letter, H, E, C, and G, we got 3 billion of those. 3 billion of those combine to form 20,000 genes. 20,000 genes are responsible of our biology. Any changes in those 20,000 genes are mutation, responsible sometimes for diseases. but sometimes without any change, you will still have the disease because of epigenetic changes. They are not always pathological. They're important for specialization of our cells. They're important for gene balance, for example, in a woman. They're important for us just being, because there are some genes that will be expressed only because they come from the mom or only because they come from the dad. Unfortunately, those methylation can also lead to pathology like in cancer, can also lead to some pathology like in malnutrition, can lead to some transgenerational effect of things that we are still grappling with, like trauma or alcoholism. But there are also another element that are beyond genetics, that are environmental and life changes. We alluded to this yesterday, malaria and sickle cell mutation. People that study evolution of malaria think that malaria have always been there for very, very long. How many long, we don't know. But people that study sickle cell disease, up to recently, up to 3 years ago or 4 years ago, we thought sickle cell disease was a mutation that evolved independently in 5 different areas of the world. 4 of those areas in Africa, and 1 of those areas in India, in response to malaria infection. Because if you have the sickle cell mutation in 1 copy, you don't have severe malaria. And our own work from 3 years ago, we found in Sudan all the signature of the 5 locations in a single country, what we call haplotype. All the haplotype was present in Sudan. And we put the hypothesis that sickle cell disease have evolved in a single place, and that place is in Sudan, probably, in Africa anyway. Because to have all the mutation, all the haplotype in a single place is only due to one possibility. Either you have a population contraction, everybody traveling and live in Sudan, or a population expansion, everybody moving from Sudan and populate the rest of Africa and the world. And population expansion is the most likely hypothesis, because Bantu expansion, human migration out of Africa, went through Sudan and Oman, the nowadays Oman. But of course, that was very challenging and a bit audacious and a bit arrogant to say that, because for the past 50 years, everybody knows his 5 origin. And we were absolutely right, because one of my friends, Charles Rotimi, showed now last year that effectively sickle cell evolved only in Africa. And it's evolved exactly 7,000 years ago, and get fixed in the population 5,000 years ago. So if we put that into human anthropology and human civilization context, it means that during the ancient Egyptian time, sickle cell was already there. So if we were able to take DNA from mummies and screen them for mutation, we should be able to see at least one of the mummy with sickle cell mutation. What I'm trying to illustrate here is that our genome evolved according to the environmental pressure. You change the environment, your genome will evolve. What is happening in West Africa is illustrated here. The sickle cell mutation lead to a type of hemoglobin called S. Another mutation here, the very same site of sickle cell mutation, lead to another type of hemoglobin called hemoglobin C. For example, in sickle cell disease, glutamic acid is replaced by valine. For hemoglobin C, is replaced by lysine. So very, very same site, but different mutation. But people that will have a double copy of S will die. People that have a double copy of SC don't die. So that's probably the perfect mutation to resist to malaria. What does that mean in terms of evolution? It means that if we don't control malaria, and if we are able to live for the next 1,000 or 2,000 years, or if someone in this room hide himself in a very, very, very powerful, I don't know, cupboard, and come in 5,000 years as a geneticist and test people, the S mutation will have been replaced by C. And that transition is happening in West Africa, where we are having more and more C and less S. Because this is a mutation that allows us to resist to malaria without killing us. Environment change our DNA, also change the way our DNA are expressed. Another way to change the gene expression and also is the nature. We spoke about monozygotic twins that do not express the same way. When they are discordant, when they do not show the very same trait with the same environment, that means that it's probably environmental but not genetics. This is one example. When they are concordant, that means they have the very same gene expression and show the disease the very same way. That means that that disease is likely to be genetic. That's the way we try to distinguish nature to nurture methodologically. Another way to distinguish nature to nurture is adoption study. Two parent, two sheep, same family, same close genetic composition. You put them into two different environments. Will they develop the very same disease the same way? If the answer is yes, it's likely to be genetics. If the answer is no, it's likely to be environmental. Behind this concept of what is genetic and what is environmental, there is a concept called heritability. The most important is to realize that it's a statistical concept that allows us to look up the proportion of what is genetic and the proportion of what is environmental. For example, schizophrenia, we know schizophrenia has to be genetic somehow. It has 85% heritability. Your schizophrenia runs into family. It will not run into family in a Mendelian way that can be evaluated statistically, but we know is highly likely to run into family up to 85%. Asthma is exactly the same. Many people will have asthma, but they will have one parent or one sibling that have also asthma. Spina bifida malformation is high blood pressure, but the essential one, the one that is very severe, 60%. Diabetes mellitus is a little bit less, 40%. 60% of the occurrence of diabetes is due to the environment. So even if you have predisposition from your parents and you change your lifestyle according to this risk, you're not going to develop it. That's exactly what it means. But for schizophrenia, whatever the lifestyle that you have, you are likely sometime to develop it because it's not something that you control. So that the most common malformation that we have in human, the heart malformation, is also involved genetic and environment. Heart disease, the one that affect our heart, also involve genetic environment. Hypercholesterolemia, we spoke to it. High blood pressure. High blood pressure is an interesting one. The study are not very, 60% is what we call essential, the difficult one to treat. But most high blood pressure that we have only have about 20% irritability, which means that if we control our environment in terms of stress, we are highly likely to control our capacity to develop high blood pressure. In fact, if we are likely to live up to age 90, we will have high blood pressure. Because the human body, the way it was designed was not designed to live that long initially. But now I think we have to live a little bit longer. I think estimate now, think that in the next 20 years, some people will live up to 120 years without any problem in the next 20 years because of the change in our capacity to control it. That also means that we need to think carefully how we're going to manage, for example, insurance, for example, retirement, because we will have to live much more longer than planned. But we will also live much more longer with our physiology that will change. The earlier human will live 40 years, but now we live in average 80, 85, 90, 100. That means that from that age, our blood vessel becomes stiff. And whether we like it or not, whether it's genetic or not, we will develop like high blood pressure. So it's not necessarily genetic, it's mostly due to the environment and the way our biology is designed. But this one is highly likely to be more genetic than environment. If you change your diet a little bit, if you don't visit McDonald's, I have nothing against McDonald's, but by the way, but it's the best example to take. You may not develop it. Obesity definitively have a good proportion of environment. This one I don't like to show it most, depending on the audience. There is some level of concordance of monozygotic twins of more than 60%, which means that alcoholism has some level of genetic component. If you come from a family where people like to drink, you are highly likely to drink a little bit more than the average. I will not show this, for example, if I've given the lecture for a wider audience, specifically in the Western Cape, because one of the major problems you have is alcoholism. And people will take it as a pretext. It's in my genes. Okay? Even if they can modify it, but they will say it's in my genes. That professor said. But in reality, it's been shown based on study that there is some level of genetic composition, but it makes sense somehow because remember, schizophrenia have a strong genetic composition and irritability. That is also the same for endogenous depression, depression endogène. Alcohol, in fact, is an antidepressant at a moderate dose. So maybe what we are seeing here as irritability of alcoholism might be also irritability of some type of depression. And that people try to control by drinking a little bit of alcohol. Schizophrenia and bipolar disorder, we've said it, is strong genetic composition, minimal environmental influence, Even though they can be managed very, very well now, a very good colleague of ours that is probably one of the strongest advocates of bipolar disorder that he has himself, was a dean at a medical school, in one of the medical schools in the US. But he knew very well that he has the condition, and he tried to use the condition in his advantage. In bipolar, you have two phases. You have a manic phases, where you want to do 1,000 things at the same time, and you have a depressive phases. The depression phase, unfortunately, happen at night usually, so it will happen at home, while the manic phase usually happen during the daytime when you are at work. So people will say at work that you are highly productive. But at home, your family will suffer because they have to control your depression. But he knew it very well, and the way he managed it is that he would try to be as productive as possible. From his own word, he will say, I will design a research in the morning, perform it in the noon, write the paper in the afternoon. And in the evening, my wife will suffer, because the wife will control the depression phase. So what I'm just trying to say is that these conditions today are manageable, and they can be even compatible to a successful professional life. It's no more like a curse that cannot be controlled. It's possible to live with it, specifically the bipolar one. Schizophrenia is a little bit more difficult, but it's possible to manage. Colorectal cancer, that is also a very common cancer in Europe and in America, less common in this part of the world. Associated in Europe and America because of high fat and low fiber diet, have a very strong genetic composition, but I'm not very convinced that it's lower in Africa. The only reason I'm not very convinced is that those studies have not stratified very well on age and life expectancy.